In this video, we will try to solve problem on the process of phosphorylation and protein structure. So during the process of phosphorylation, generally ADP is converting into ATP that occurs through energy metabolism. Comprises of kind of phosphorylation process are oxidative phosphorylation, maybe substrate level phosphorylation, or photophosphorylation, means it occurs in the presence of light, especially in plants. So this ATP can also be formed from ADP through an action of adenylate cyclase. So we all know the function of kinase enzyme, right? It contains crystal structure. So determination of protein structure sequences of adenylate cyclase, that is given, and it basically contains two terminal regions, C terminal and N terminal. So here particularly C terminal region sequences are given, and this is the whole sequence of total 15 to 18 amino acid sequence are given. So among this, based on this information, we have to find out that kind of conformation of this sequence may be. Option A says that it is an alpha helix that is not amphipathic in nature. B says that amphipathic helix may be present. Option C says that it may be leucine zipper, or option D says that it is an beta hairpin model. So in option C, leucine zipper and beta hairpin models are in two-dimensional form. So option C and D is directly excluded because here only a sequence of amino acids are given. So we have to just find out that this sequence are amphipathic or not. So what is the meaning of amphipathic? That we know that it contains hydrophilic and hydrophobic both amino acids. Means hydrophilic, that means polar amino acids, and hydrophobic means non-polar amino acids. Means it do not attract water or any other polar solvents closer to them, right? So in this hierarchy of sequence, we can see that valine, it may be aspartate or else aspargine amino acids, whereas threonine, aspargine or else aspartic acid, and again threonine, these are highlighted amino acids. We all know that if you remember what are hydrophilic and hydrophobic or else non-polar polar amino acids, then these amino acids are particularly polar amino acids. And rather than this, valine, phenylalanine, serine, cysteine, histidine and leucines, these amino acids are non-polar amino acids. So this sequence, whole total sequence of protein structure sequence, is in combination of both polar as well as non-polar amino acids. So there are chances that it is containing amphipathic sequence. So we have we have to confirm that among option A and option B, which one is correct. Option A says that it's an alpha helix that is not amphipathic. Yes, alpha helix is present in a sequential form also, but it's not amphipathic. But in the sequence, it's a combination of both polar as well as non-polar amino acids. So option B says that amphipathic helix is present, no matter it's an right-handed or else left-handed helix, right? So option B is the right answer of this problem. So if my videos are helpful to you, please subscribe to my channel. Thank you. Have a nice day.